going to discuss about post-transcriptional modification. Post-transcriptional modification or co-transcriptional modification is a set of biological process in which the premature RNA, which was the is the outcome of transcriptional process, is changed into a mature RNA through a set of chemical processes. There was a quite difference between prokaryotes and eukaryotes where the PTM modification takes place. In prokaryotes, the post-transcriptional modification was not suddenly found because the mRNA, which was directly transcribed from DNA, was immediately used in the translation to get proteins. PTM modifications was occurs only in the nucleus of the cell. Most PTMs are responsible for changes in rRNA, tRNA, and other special RNAs like sRNA and SNO-RNAs. This PTM modification was achieved by 3 steps. First was 5N capping. Let me see about 5N capping, which is known as 5' terminal cap structures, in which the premature RNA, whereas the triphosphate group was added at the 5' was capped with the 7-methyl guanosine triphosphate, which is also carried out by a specific steps in which the phosphate enzyme removes the phosphate groups at the 5' of the premature RNA and adds the 7-methyl glycosine triphosphates with a enzyme known as guanyl transferase, and it leads to the methylation at the 7' position of guanine through a methyl transferase from an enzyme from a compound known as 7-adenosyl methionine. This was known as CAP0, whereas the methyl group was added at the 7' position of guanosine. Next, the methylation occurs at the 2' end of OH group of the ribose sugar. This was known as CAP1, and further CAP3, CAP4 occurs at the adjacent ribose. This cap protects the 5' end of the primary RNA transcript from the attack of ribonucleases. Some of the abundant different cap structures are: yeast will proceed to cap0. Some slime modes are mainly cap0, but 20% have cap1 structure. Messages of brine strain and sea urchins will always terminate at the cap1. Most mammalian groups always possess more than cap1, 2, 3. And in cytoplasm, cap interacts with the EIF4F to initiate translation process. And rRNA, tRNA are not capped always. Most snRNA contains the trimethylglycosine as a cap. The second step was polyadenylation. This is a process where at 3' end of the RNA molecule involves cleavage of its 3' end and then the addition of adenosine residues to form a poly A tail, in which the polyadenylation sequence, which was known as AAU3 adenosine, and which was followed by GU-rich sequences. This aids in the polyadenylation process, and there was some specific protein binding proteins known as CPSF, CSTF, and 2 cleavage factors were also helps in cleavage of nucleotides, in which CPSF always attached with the signal sequence and CSTF attached with the GU-rich sequence. Cleavage factors attach at the cleaving sites and cleaves the nucleotides and aids to the attachment of PAP, polyadenylate polymerase, which always initiates and aids in the sequence and to the addition of adenosines. There were more than 200 adenosines are attached at the nucleotide sequence to get a poly A tail, which protects the 3' end from ribonuclease digestions. The last step was known as splicing, in which the introns, the non-coding regions, are removed through an enzyme known as spliceosomes, and also it contains snRNAs, which was attached at the splicing sites and removes the neutrons to give a mature RNA. This was transported into the cytoplasm and aids in the translation process. Some of the significance are: translation always uh, efficiently initiates the translation process and prevents the exonuclease digestion, and regulation of quality control of RNA in protein synthesis, and aids in the export of mRNA from nucleus to cytoplasm through a nuclear pores. They also help to protect the mRNA from degradation. Thank you, sir.